pleasure to introduce the first speaker of today, Dr. Sanne Janssen. She started her journey with a degree in biology at the Radboud University in Nijmegen in the Netherlands, and then continued at Radboud for a master in medical biology, to then move to the McGill University for a PhD in pathology. After graduation, she stayed in Canada, moving to the University of British Columbia for a postdoc in the lab of Matthew Lorinx, where she's working on the interplay between chromatin and transcription during early embryo development, about which we'll hear more in her talk entitled Dissecting the Polycomb-H3K37 dimethylation axis in transcriptional regulation. Sanne, we're very glad to have you here, and the floor is yours.
Great, thank you. Okay, let me get set up here. Perfect. Okay, thank you so much for that introduction. And thank you for the organizers for giving me this opportunity to give a talk. My name is Sanne and I'm a postdoc at Lorinx's lab. So in the Lorinx lab, we are interested in gaining a better understanding of the complex crosstalk that takes place between a number of chromatin modifiers and the marks that they deposit, as shown on this slide. For the talk today, I will be focusing on this bottom corner, so the crosstalk between Polycomb and K36 dye, and how this crosstalk impacts the transcriptional landscape. So as a heads up, this is by no means a finished story, and there's still many unanswered questions that remain. So why am I interested in these chromatin modifiers? Well, one of the reasons is that they are all identified as being mutated in neurodevelopmental syndromes, including the ones listed here, as well as in a number of cancers, actually. So by obtaining a better understanding of how crosstalk between these modifiers and their marks impacts the transcriptional landscape, we hope to gain insight into the molecular basis that underlies them. So let me start with briefly introducing our first two players of today. So first we have PRC2. So PRC2 stands for Polycomb Repressive Complex 2, and it consists of three core subunits, Sus12 EED, and a catalytic subunit EZH1 or EZH2, which is responsible for the deposition of three methyl groups on histone H3 at lysine 27. So in the cell model system that I'm working in, EZH2 is actually the major catalytic subunit. So then on the other side, we have NSD1, which is responsible for the deposition of K36 dimethylation. So it was shown that the EZH2 subunit of PRC2 contains a so-called H3K36 sensing pocket. And when K36 is methylated, it inhibits PRC2's catalytic activity. And so in agreement with these observations, it has been shown now by many groups that loss of K36 dimethylation leads to a broad gain in K27 trimethylation. Here on the right, I'm showing an example of this from our lab from a previous postdoc, where I hope you can appreciate here in the highlighted area that when he knocked out NSD1, this was done in mouse male germ cells, you see a loss in K36 dye. And in the same regions, he observed a gain in K27 tri. Other groups have also shown that this gain in K27 trimethylation can also occur in de novo sites. Another observation was that when he knocked out NSD1 in these cells, that there is altered transcription, as shown on the left here in this MA plot. Intersecting this gene expression data with ChIP-seq data for K27 tri, as shown here on the right, revealed that a large number of these downregulated genes show a gain in K27 tri, in this case over their gene body. So all these observations together led us to ask the question, is this aberrant gain in K27 trimethylation that we are seeing actually responsible for the transcriptional repression that we observe in NSD1 knockout cells? And so in other words, we're asking if the role of NSD1 in transcriptional regulation is polycomb dependent. To study this, we're making use of naive embryonic stem cells, NSD1 wild type and knockout, that were isolated from the mice used in the previous study. And we validated by Western that these cells are lacking K36 dimethylation and show this aberrant gain in K27 tri. Next, we performed RNA-seq and ChIP-seq on these cells, and I'm showing that here in this heat map. So focusing on the left column here is the gene expression changes, and in the bottom box, I am highlighting the downregulated genes. Now looking at the ChIP-seq data, I hope you can appreciate that when we compare wild type to knockout, we see this large gain in K27 trimethylation, and in this case, we're looking at the transcription start site and its flanking regions. So we wanted to now ask the question, if we remove this K27 trimethylation, do these genes become de-repressed, or are we rescuing this gene expression profile? So to that end, we obtained PRC2 Degron cell line from the closed lab. So what these cells are is that they have one of the PRC2 subunits is 12 fused to a Degron tag. And when we treat these cells with a compound called DTAG13, we're able to degrade those 12, leading to a depletion in K27 trimethylation. So I used these cells, I used CRISPR to knock out NSD1 in these cells and validated by Western the loss in K36 dimethylation. In addition, I validated that when we treat these cells with the degrader compound, that we are seeing a loss in K27 tri. Next, we performed RNA-seq to actually look at what happens to gene expression. So in the left scatter plot, plotting expression in wild type compared to knockout cells, we identified the downregulated genes in red. And now looking at these genes in violin plots, so here going from wild type to knockout, you can see these genes are downregulated. Then looking at what happens when we now degrade PRC2, we were kind of surprised to see that there is actually no de-repression or rescue of these downregulated genes, indicating that K27 trimethylation is not responsible for the downregulation of these genes. So while at first this was a bit surprising, we then went to the literature and we saw this paper from the closed lab, basically showing that PRC1 is more important for transcriptional silencing compared to PRC2. So before I dive into this data, let me first briefly introduce PRC1. So PRC1 stands for Polycomb Repressive Complex 1. And its catalytic subunit, either ring 1A or ring 1B, is responsible for the deposition of a ubiquitin group at lysine 119 of histone H2A. I will be referring to this as H2AUB just for simplicity. In the cells, so in the ES cells that we're working with, ring 1B is the major catalytic subunit of this complex. So PRC1 has an interesting positive feedback loop with PRC2, whereby when K27 tri is deposited in the genome in specific sites, it can lead to the recruitment of specific PRC1 subcomplexes, resulting in the deposition of H2AUB. And on the other hand, H2AUB deposition can lead to the recruitment of specific PRC2 complexes, resulting in the deposition of K27 tri. Okay, so coming back to this observation from the closed lab. So basically what they found was that the degradation of PRC2 with these cells that I just introduced before did not lead to any de-repression of polycomb target genes. So in addition, they had this other Degron line, which is the PRC1 Degron cells. So in these cells, ring 1A, so one of the two catalytic subunits, is constitutively knocked out, and ring 1B is fused to a Degron that can be induced by the addition of auxin to the cells. So they used both of these systems, induced these Degrons for 2 hours, and did not observe any de-repression upon PRC2 degradation, but did see large-scale de-repression upon PRC1 degradation. We thought this was interesting, given that we also don't see any de-repression with the PRC2 Degron system, and we're wondering if maybe PRC1 plays a role in our system, given the feedback that I just explained before. So what we did is we wanted to reproduce this data, but instead of degrading for only 2 hours, we wanted to degrade for longer, as K27 trimethylation is known to be a more stable mark than H2AUB. We degraded for 48 hours, and just showing here the Westerns confirming the loss of the respective marks, and performed RNA-seq. is shown here in the scatter plots. So similarly to the observations by the closed lab, we found that PRC2 degradation led to very minor transcriptional changes, especially compared to PRC1 degradation, where we see this massive gain in expression of a number of genes. Okay, so this made us wonder, if PRC1 might play a role in the downregulation of genes in the NSD1 knockout cells. So to answer this question, we used these PRC1 degron cells and by CRISPR knocked out NSD1 in these cells. So that is what I'm showing here, the validation by Western of the loss of K36 dimethylation. Performing RNA-seq in these cells, we were able to identify a number of downregulated genes. And similarly to what I was showing before with this violin plot, is we can see the downregulated genes here from wild type to knockout. And then when we induce the degron in these cells, we this time actually do see derepression of a number of genes, as shown here by this shift in the violin plot. Okay, so if H2AUB or PRC1 is indeed playing a role in the downregulation of these genes, we expect to see a change in H2AUB at regulatory elements in these NSD1 knockout cells. So to address that, we first just performed a Western as shown here in the top to look at the global levels of H2AUB. And in contrast to K27-TRI, which shows this clear gain in this mark, we did not observe any global changes in H2AUB. Next, we performed ChIP-seq and looked specifically at the promoters of the downregulated genes. Looking here in green at H2AUB, you can see that there is also no change in H2AUB at the promoters of these downregulated genes, again, in contrast to K27-TRI methylation. So the next logical step for us was to look at another regulatory element, specifically enhancers, as there's also been a couple papers that have shown enrichment of NSD1 as well as K36 dimethylation at enhancers. So to do that, we mined H3K27 acetyl high chip data from the Greenberg lab, which is specifically generated also in naive embryonic stem cells. And we use this data to annotate enhancers to NSD1 downregulated genes. We were able to do that for 267 genes. Here I'm showing an example gene. So in blue highlighted is the enhancer, and in yellow is the promoter. Next, we performed K27 acetyl chip in these cells to identify enhancers of downregulated genes that are being decommissioned. So that is the data showing here, and you can nicely see the K27 acetyl peak at the enhancer disappearing in the knockout. So about 1/3 of the downregulated genes that have an annotated enhancer showed this K27 acetyl loss. Okay, so what is happening to the polycomb marks at these enhancers? So we quantified these over the enhancers, and what we observed is that there is a very minimal gain in H2AUB over the enhancers of the downregulated genes in the knockout. But again, when you compare that to K27-TRI, there is a really clear gain in K27-TRI over these enhancers. So we were wondering, is this minor gain in H2AUB that we're seeing, is that actually meaningful? And so when we look at a genome browser screenshot of our data, here on the left, the enhancer, you can see how this is a very minimal H2AUB gain. What we also saw when we looked at this and what stood out was the H2AUB that is actually present in the promoter already in wild type cells. So this raised the possibility that the transcriptional derepression that we're observing following PRC1 degradation is actually a consequence of the loss of H2AUB at the promoters of downregulated genes. And so looking at the expression profile of our example gene here, you can see that even that in wild type cells treated with auxin, so with PRC1 degradation, there is also derepression. And so quantifying H2AUB over the promoters of our downregulated genes in wild type cells, comparing all genes to those that are downregulated, you can see that H2AUB is actually enriched at the promoters of our downregulated genes. So this indicates that it's not H2AUB that is causing or that is responsible for the downregulation of genes in our NSD1 knockout cells. So to conclude, what I've found so far is that when we deplete PRC2 in NSD1 knockout cells, we do not see derepression of genes that are downregulated, indicating this massive gain in K27-TRI that we and many others have seen is not responsible for this downregulation. On the other hand, we do see derepression of genes when we degrade PRC1, but this seems to be more so because we are depleting H2AUB from promoters of the downregulated genes that where H2AUB is already present in wild-type cells too. So that is depicted here by this inhibitory arrow. So for this, the conclusion is that the gene downregulation in NSD1 knockout cells is likely not mediated via polycomb marks. So what we also observe is that a subset of genes downregulated do show a loss in K27 acetyl at their putative enhancer. So does NSD1 and or K36 dye somehow promote enhancer commissioning? This raised the question if there might be also a catalytic independent function for NSD1 in enhancer commissioning. And the reason I'm bringing this up is actually last year, there was a paper from Christian Helene's lab that showed that NSD1 promotes enhancer activity by stimulating RNA pol-2 pos release. So this is one way that potentially NSD1 in a catalytically independent way can promote enhancer commissioning. Another open question that still remains is if NSD1 can restrict other repressive histone marks. So these are questions that we are currently working on. So with that, I would like to thank everybody in the Lawrence Lab, specifically Matt for his great guidance, the funding resources that helping me do this research. And if anybody is interested, the Lawrence Lab is currently looking for postdocs. So thank you all for listening, and I'm happy to take any questions.
Fantastic, Sanne. Very, very elegant and nice talk. While we wait for we already have a question in. Go ahead.
So that was a really great talk. I really enjoyed that. I was struck by your observation. People have seen similar things previously that like PRC2 seems to do so little for gene expression in the naive MESCs. And I was wondering, do you think this is unique to the MESCs, or would you see something similar if you did the experiment in a differentiated cell type?
Yeah, that is a great question, Ben. So I wish I could have done the experiment, or I actually tried to do it in by differentiating these cells to epiblast-like cells. But unfortunately, the NSD1 knockout does not support that differentiation. So I would have to do that with other cells. It might be that that it is different because the distribution of polycomb, of course, changes when you differentiate cells. But yeah, I have not done that myself.
Thanks.
Yeah, that was one of my questions as well. That was a very good point. So I was wondering, do you think that the deposition of K27-TRI is just a result of the lysine being available because it's not acetylated anymore?
So
Or
you're
is there anything else?
You're asking if the gain in K27-TRI, why that is happening?
Yeah, so if this actually just the default that is installed on the histone because it's not acetylated anymore, or if there is anything else that you think is at play there.
Yeah, so I think that PRC2 is just a very, I think the English word is spurious enzyme, where it basically will deposit K27 trimethylation wherever it can, and that NSD1's mediated K36 dimethylation, as well as we know K36 trimethylation does the same, where it really restricts where PRC2 can deposit its mark. Yeah, so it's really restricted by other modifications.
And partially connected to that, from your initial Western on the K27 methylation increase in the NSD1 knockout, the increase is really substantial. Have you checked whether that is only happening on gene bodies or also around scattered in the genome? Because that level of increase to me would suggest that there's also other targets that are acquiring, other locations that are acquiring K27 tri.
Yes, so it is not just at gene bodies. We also see it at promoters and flanking sites. When I just look in the genome browser, it is nearly everywhere where it is not being excluded by something else. So yes, that is also at enhancers at all kinds of places.
I think Ben has another question.
Yes, so just following up on the acetylation a bit more, have you looked at any other sites other than K27 acetyl? And why I'm asking, I'm thinking of the enhancer decommissioning that you might be seeing, and the increase in K27 tri and the knockout could actually block K27 acetyl just as the same mark. And so if you looked at something else like K18 acetyl, it might point to a more general decommissioning of the enhancer beyond just the K27 acetyl.
Yeah, it was a great point. I have not yet done that because this is relatively new data, but we will definitely be doing that, looking more into it. Yes.
Great. So if there's no other question, thanks a lot again, Sanne. And I'll pass it on to Jackson for the next talk.